And so I find that very inspiring. I got to know Alexis over this week. So Alexis, through a funding program with IVADO, there's an IVADO INRIA program. So this is what allowed Alexis to come here. And I'm also going to Montpellier in a few weeks to visit Alexis' team. So it's a real pleasure to have you here. Thanks so much for agreeing to give a talk, Alexis.
Yeah. So yeah, thanks a lot for the invitation and for the organizers, and yeah, special thanks to Etienne who invited me here for 2 weeks in Montreal. Yeah, so we have already been talking a lot about the motivations during the previous panel session, but yeah, for those who did not attend and those online, just in a nutshell, one of the key challenge that motivated the work that I'm going to present today is what we call the taxonomic gap or taxonomic impediment, which is the fact that only highly skilled experts are able to identify species accurately. And this has been recognized as a major obstacle to biodiversity monitoring since already the Rio Earth Summit in 1992. And so, of course, this is becoming more and more critical, in particular because we have now strong engagements towards biodiversity conservation at both the global and the local level. And yeah, so in particular, some engagement for 2030, so which is in 5 years now. So yeah, this taxonomic gap was the main reason why we created PlantNet 15 years ago. And yeah, so PlantNet is a citizen science platform that is centrally relying on artificial intelligence to help people identify and monitor plant biodiversity with their mobile phones. So now it's quite a well-established platform with about 30 million people using it. And all those people collect data, and those data are shared in open data infrastructures like the GBIF, which is the world's largest one. And there is a rich community of users, of contributors. So some are really amateurs, but some others are professionals in agriculture, conservation, education, and so on and so forth. So yeah, despite this success, plant identification still remains a difficult problem. So why is that? So the first big problem is that there is a huge number of species, so 400,000 species according to the last estimates. And so if all those species would be very different, that will only be a scalability issue. But yeah, unfortunately, this is the contrary. And so you have a huge, a very low interclass variability. And yeah, this makes it a very extremely challenging fine-grained classification problem. And on the opposite, you have a very high intraclass variability. So first, because the plant is composed of several organs, and these organs have a high plasticity across years and across seasons. So something that can help is taxonomy. So the fact that life is organized in a hierarchical manner, and this is something that naturalists are using from centuries now to classify species. Yeah, and so in general, species that belong to the same genus parent nodes, they are more likely to be similar. But this is not always true, and you can see on the right that for some genus, the species can be very different visually speaking, and so you cannot have a general easy rule to use in your models. So then, of course, we have the well-known problem of the long tail distribution of the data. And to illustrate it, we can cite this study of Nigel Pittman, from Field Museum, to which we participated. So he aggregated 25 of the biggest online databases with images of plants. And yeah, he matched that with the list of all species in North and South America. And yeah, he realized that only 47%, no, I mean, the opposite. So 47% of the species had not a single image in all those databases. So it's not even a long tail, it's just that they don't exist in pictures. They are herbariums or somewhere else. And of course, there is also a very large number of few shot species. So more than 80% of them only have few images. Yeah, what is less known is that it's not only a long tail, it's a long tail in the long tail. So the species follow a long tail, the genus follow a long tail, but there is also the species inside a genus that follows a long tail. And so you can model such distribution with a joint zip distribution like we see here. So yeah, one of them is at the genus level, the other one, the species inside the genus, and the product of those two zips creates the final zips you see at the species level. So, but indeed, there is some intrication before. And this distribution makes that some few shot species are more difficult than others to recognize. So if you have one few shot species, like the one bottom left, in a genus where there is another similar species with a lot of images, the model can capture the visual features quite easy because it can capture that. But for other genus, like the one on the right, all the species are few shots, and so those ones are much more challenging to identify. So yeah, more fundamentally, in the end, you have prediction uncertainty, and you have two types of prediction uncertainty. So the first one is due to this ambiguity. And so, I mean, if you take two species like the one on the left, some of them can have really the same type of leaf. That's not the criterion that is used to distinguish them. So if someone only take a picture of the leaf, you cannot distinguish the species. So even an expert cannot distinguish the species. So this is an irreducible uncertainty. So I mean, you can have an infinite number of training samples in your data sets. Your model will perfectly learn this ambiguity with no uncertainty, but it's ambiguous, all right? And on the other side, you have model uncertainty, which is more due to lack of knowledge, to the errors you do during training. And so, yeah, this one, you can try to improve it with better models and, yeah, of course, more training data. Yeah, and unfortunately, it's not finished. You also have label noise. Yeah, because images are labeled by more or less expert contributors, and even the best experts make errors because, yeah, first, taxonomy is kind of living things, meaning names change, synonyms over time, and yeah, and depending on how they identify the species, they can make error. If you only provide them a picture, it's just a part of the information. So that, I mean, it's not like identifying the image in the field, and they will make error. We measure that, so even best experts can have 70 or 90% accuracy in the task of identifying pictures. Right. So in the end, usually you need to predict sets of species and not only one top species. And so you, instead of having your hypothesis space that is just 400,000 species, no, it's the hyperset of the set of 400,000 species. Yeah, of course, we have less species in reality in the systems, but yeah, that's true to say that. And so, yeah, your classifier becomes a set-valued classifier, so you have to pick up one combination of species and the most appropriate one. So the easiest thing you can do is top K classification. So I just return the K most probable species. So you can do that easily with the softmax and your cross entropy. But you can also try to do better by optimizing the top K error itself. And so, yeah, this is a work one of my PhD students worked on, Camille Garcin. So if you look at the top K error, it's just a 0-1 loss. So it's 0 if your class is in the top K, it's 1 if it is not, but this is not differentiable. And so if you want to use that in your model, you need to smooth this objective. And so we were doing that with what is called perturb optimizers. So simply you will add some Gaussian noise to your logits. So it's not logits in this case, but yeah, that's equivalent scores. And so you will be able to compute the expectation of a species being the top K. So it would turn that into a probability of being the top K. And yeah, so basically this creates a smooth version of this top K error. And yeah, you can have some gains on the top K classification. So, and you also add an imbalanced version of that that provides some pretty good scores. Yeah, so, but the problem of top K is that it's not adaptive. So what you would prefer is when you are sure about the species, you only return one species. And when there is a lot of ambiguity, you return many classes. So this does not work with top K. So one thing we have been working on, so it was another PhD student, teacher on L'Oreal, is average K classification. So here the idea is that you will return K classes on average, but the number can change for a particular sample to another one. And so if you formalize this objective, you will see that the optimal theoretical solution is just to threshold the conditional probability of the species. So it's just the threshold, and you just have to find the optimal threshold that gives you K classes on average, all right? And it also showed that the cross entropy is well calibrated for this problem. So it's called average K calibrated, which means that in the infinite case, sorry, there is a property that is called a strongly proper loss that has the cross entropy. So which means that it really tends towards the real conditional probability. And so, yeah, if you just use the softmax and cross entropy, if you have enough data, you can just threshold and you have some optimal solution for that. Yeah, sorry.
Okay. I have a question. This might be a very naive one, but like imagine you have n classes. Can't you predict just the probability of belonging to each of these n classes, and that's it? And like if the probability is equal, is like centered on one of these classes, it means like we're sure about the species, and if it's scattered.
Yeah, exactly. That's what this equation is saying. Okay, yeah, I mean, that means that yeah, if you just have an estimator of the probability of each species, you threshold it, and you have the optimum average K classifier. Okay. But this is not so easy to prove theoretically. So I mean, there is a lot of maths behind. Yeah, so then it's in the infinite case. So cross entropy is optimal in the infinite case, but you can also try to do better in the real cases where you only have some samples. And so, yeah, Camille Garcin has been working on some two-head loss functions where there is one branch using the cross entropy, and so this gives you the conditional probabilities. You threshold, and you use the pseudo labels for another head with binary cross entropy, and and you ensure that on average you have K species during the training, and so this allows you to have a few improvements, so not big ones, as you can see. And the idea is that in the end you predict set sizes that are larger for species where there's the more uncertainty. So a last piece of work in this thematic is conformal prediction. So this is a topic that interests some researchers in my team, like Joseph Salmon. And so he has been working with Tiffany Ding from Berkeley about that. So if you look at the classical conformal predictor, it's exactly like average K classification, except that you will calibrate the threshold on a calibration data set. So you have labels. So average K, you just control the set size on average. With conformal prediction, you will really control the errors, all right? So the base one, it's only a global threshold on your softmax. And then there is some variants that are class-wise conformal predictors. And so in those methods, you will learn a threshold for each species, okay, to control the error for each of the species. But when you have a long tail distribution, that does not work at all, because you will predict a lot of species for when it's uncertain. And so in the end, the average sets are far too big. And so what Tiffany has been working on is an interpolation of the threshold, the global threshold, and the threshold for each class. So it's just an interpolation, but that allows you to find the best trade-off between using a global threshold that is usually good for the long tail because you have too much uncertainty on the long tail, and the class-wise threshold that are easy to estimate for the most frequent classes. And so if you just interpolate between those two thresholds, you can find some better trade-off in terms of set size versus coverage. Yeah, if you are interested by this kind of topic, we are sharing a data set that is called PlantNet 300K. That was published in the RIPS dataset track. And the key idea is that it's a subsampling of the full PlantNet database, but at the genus level to preserve the long tail in the long tail, which is very important. And as far as I know, it's the only one good like that. And it changed the performance. We have some experiments on Cypher that show that. And in the end, it's preserved the long tail, the classical long tail of PlantNet. So it's basically the same distribution at the species level. Yeah, I'm not going to talk about this problem today, but there is also this plant cleft challenge that we organize, where the idea is more a multi-label classification problem. So you have to predict all the species present in high-resolution images, but the training set is still PlantNet data, so individual plants, and so there is a big domain shift, and the performance are very low, so plenty of research opportunities there. So now I talked more about PlantNet. So how do we implement all that in the platform itself? All right. So the key concept of PlantNet is what we call cooperative learning. Which means that the data that is used to train the model is entirely produced, revised, and validated by the community. All right. And so they take pictures of plants, and the more experts are validating them, and then this enrich the system, and we will see how this works more closely. So when there are different labels for the same observation, we are using a weighted majority voting rule. So each user has a weight in this majority voting rule, and the weight is expected to reflect the user expertise, right? So you want to give much more weights to experts and much more weights to experts. Yeah, please. It's appuie long, faut appuyer longtemps. Ouais. Sorry, you have to. Can we read it?
So the question is, how are you eliciting the user label? So you could ask the user, you know, what's the species? But you could also ask in between these two, which one's the most likely. You could have. There are many ways to elicit a user response. I can try different ways.
And yes, I will talk after how we suggest some contents for collecting the labels. Yeah, so this is later in the talk. Yeah, so and the key question is how to estimate the weights of the users, and therefore we are using an expectation maximization algorithm. So the idea is that the algorithm starts with equal weights for everybody, so it's just a voting rule. This allows to have a first estimate of the most likely species for each of the observations, and some thresholds on those estimates to only keep the ones that are highly confident, so we are sure about them. And then the weights are computed based on the number of correct species of each user. So it's not their global probability of success, it's more the number of species, because we realize it's much more informative about user expertise than just being able to label easy plants. I mean, you can have plenty of easy plants, so really the number of species is something that is more indicative about user expertise. And so you run this algorithm, yeah, so it's run every night, but it's only incremental, so we only update the new observations and new users and new things, but every night there is a new computation on all the votes to update that. Yeah, so, I mean, Of course, botanists have a high grades. That's what we expect. So this is my colleague Pierre Bonnet. So he's leading PlantNet with myself. So who has observed, so it's either observed species or validated species. So I don't enter the details, but yeah, it's more than 1,000 species for Pierre. Me, it's 300. So probably I have a higher weights because I'm not botanist, but I'm quite of biased, let's say. So I'm trying to not contribute too much in the system. That's a problem. Yeah, and when you start, of course, you have a very, very low weight, and so you have no chance to have your data appearing in the training set. Yeah, yeah, we have been evaluating this algorithm and compared it to different other algorithms. So yeah, including the 2/3 rule used in iNaturalist. So the 2/3 rule is very good in quality, but it eliminates a lot of data. So I mean, so if you have enough data, like in iNet, maybe it's not a problem. But in our case, yeah, I will show some numbers after. But yeah, but in terms of recall and precision threshold, so yeah, it's much better using an algorithm like this expectation maximization. Yeah, so. Here are a few numbers. So we have collected more than 1 billion raw observations. So it's just people trying to identify a plant. So 1 billion, so it's really noisy data. And from all this noisy data, there is some observation that people agree to share openly in the platform. So for which there is a license and they agree to put it publicly available. So, and this is only 25 million. So because a lot of people just use it to identify plants and they are not in the cooperative, yeah, or not aware, or they don't know, or they don't want. So 25 million shared observations, and when we apply the algorithm I talked about, so only this one can be revised because it's the only one appearing in the application. And so, and from those 25, we only have 18 million that are considered as valid for training the AI. All right. Yeah, so the current model in production is a Dino V2 model that is fine-tuned or unfrozen, but yeah, tuned on the 18 million PlantNet images. There are multiple heads, and yeah, and my slide is not up to date since we have 77,000 species now in the classifier, and that's 53. So yeah, sorry for that. Yeah, there is a classifier for species, one for genus, one for family. We also are working on disease, plant disease, for instance. And there is also like a classifier for reject classes and types of organs and things like that. This is important because, yeah, 1 or 2% of the queries are not plant at all. And so fungi, so a lot of people believe it's plants, but they are not. Yeah, so some tattoos. So that's real usage. Sometimes people really want to know what is the species they have on their body. So and this is very frequent. Yeah, and sometimes it's just, yeah, you don't know. People are playing with the app. I mean, that's crazy what you can have. And yeah, so it's not so easy to train these reject classes. Here it seems easy, but if you really look accurately, there is 4 images with a plant. I'll let you check, but yeah. Under the parrot, here, tree. And these are easy cases. There is a lot of cases where it's even much more ambiguous. I mean, yeah, so. Yeah, another thing we are doing to try to reduce uncertainty is to have several views of the plants. So yeah, to get a picture of the different organs. Yeah, so we ask the people to select the organ. That's, yeah, to collect labels also, but it's a way to encourage people to do close-up pictures. If you don't do that, people are 10 meters away from the plant and they just say, okay, it's a majestic, I'll just get an answer. And so, no, it doesn't work like that. You have to focus on the plant. And yeah, and we are combining the predictions using this weighted max function, which is more robust than a weighted average. So it put more weights on the picture that is the most informative in terms of probability. And there are weights because different organs are, I mean, flower is very discriminative, whereas the bark or the trunk, for instance, of a tree is much less informative. We also use some regional and thematic floras. So based on your GPS location, the app will choose a species checklist that is adapted to your country. If you take plants in the wild, if you take ornamental plants, you will be automatically switched to a global flora. But this reduces the hypothesis space, and this can improve performance quite a lot. So there is an example on the right. Yeah, you see the accuracy values when you use the global checklist or the Southwestern Europe checklist or an even smaller checklist in France in the region. Another thing to try to improve quality is similarity search. So the system returns the most likely classes according to the model, but each class is illustrated by the most similar picture to the query. So if you take a picture of the query of the buds, it won't return the same images. It will really return the images in the whole database that are the most similar to what you see. All right, and this is one of the difference with many other apps, because it really gives you something that is very close to what you see, and you can have a user control on that. So yeah, this is based on my old life when I was researcher, more on hashing techniques to speed up that. So after people have identified plants, so that's where we collect labels. So there are different ways to collect labels. So you can either confirm the AI with the risk it comes with. I will talk to you a little bit about that. You can confirm another one if like here you see visually that the second one, and maybe the model was wrong. And of course, you can also, if you are expert and if you know the species, you can contribute directly a label. And so, I mean, people contributing to the database use it like that. So there is no comparison between, I mean, this is the comparison where you see the most likely species and you have to choose, and you have the most similar pictures. And then in the whole system, in the whole platforms, you can revise observation of other users, and so there is just interfaces that allow you to suggest other names, vote on existing names proposed by others. People can vote on the quality, also on the common names. We are trying to keep this principle of transparency, meaning that all what the AI sees is what you see in the galleries. And so if you are unhappy with one species that is not recognized, please go to the gallery and look what's there. And if there is only 2 very bad pictures, you have the answer. We also have some active learning mechanisms. So like a review galleries where we put forward the most uncertain pictures according to the AI. Or the one for which there is the most disagreement between users. There is 2 types of uncertainty, and we try to put this content to be labeled in priority, and so So overall, this creates a positive feedback loop, and the more data you collect and revise, the more contributors there you have, and the more data they will validate, and so the AI will progress, and so on and so forth. So this seems to work. In 15 years ago, we had only 500 species, and now we have almost 80,000 species, and we see the progress. Please.
Just a quick question. Internally, when you get more and more data, more research grade entries has been put into the system, how do you make decisions about retraining the model to incorporate these new kind of rare species per se?
So I mean, this is usually, this is not like a workflow that train a model every 2 weeks. It's still very manual because you always have a lot of surprises with the new model. So we have data, we have test data sets that are not shared anywhere. So this is really what we use to validate a new model. And but yeah, each time the data is different, and so sometimes you don't know why you have a model that doesn't work on one of these data sets, and so yeah, you won't put it in production. And of course, we also try new architectures, and we add the disease classifier. So each time there are new things and new models we try, or new loss. So we train models in continuously. And when we are happy with a new model, because yeah, it fills all the checklist, all the checks, and yeah, we put it in production. So, and we discuss it in the technical committee, and so yeah, there is a process to say, okay, go or not go for a new model. Yeah, and unfortunately, sometimes there are some negative feedback loops, and for a few hundred species, we estimate. Sometimes you start to have an error in the training set. So two close species or an ornamental variant of a species start to be there. And so, yeah, the system will start to put some probability on it. And if you are unlucky for one particular picture, so of the wrong species, yeah, the system will return it first. And so people will validate it. And so you will have a new heterogeneous image in your gallery. And in the end, it continues, and you have really two species in one gallery. I mean, you really have two different species alive in that. So people tell us about such species, so and we try to involve them in, like, we put weights to them to be able to let them curate this kind of classes, for instance. So there are some manual curation to solve this problem, so to summarize, but it's not so easy. So sometimes you have to really reset, I mean, the species, except for the users who are absolutely sure they have the correct label. Yeah, so just a few tips about those potential negative feedback loops. So I think AI calibration is crucial. So the more accurate the uncertainty measure is, and the less bias you have from the human in the loop process, because they can really see if the AI is sure or not. AI transparency, I talked about it. Also, the AI should not become self-validating. So it's very useful to detect errors or to detect uncertain samples, but you have to avoid that, it's trained on its own pseudo-labels. I mean, it works if you do it. We know self-distillation worked, we measured it, but it's risky for those few species that are that have a negative feedback loop. And of course, we should not stop training new botanists. So, but we should not stop training new botanists. I mean, that's so important. Yeah, so now it's the more second part of my talk about how to go from plant occurrences to species maps. Because all that raw species, even if there is a lot of them, they are sparse for a particular species if you look locally, and also they are biased by observational bias, meaning that you have data where people go and not only where the plant is. So in ecology, what researchers are doing for that is what is called species distribution models. So you will use some predictors like temperature or precipitation to learn the preference of the species and then to predict where it could be. So the potential species distribution. And we have been working on such models, but more with deep learning technologies since 2018 through the PhD of Christophe Bottella. And so we were like extending the models they used in ecology with an encoder that just take input variables and learn a latent representation, and then you can apply the models they are working at. So the two findings in those models were that the more species you inject in the model, and the better the model is for all the species together. So meaning that it really worked to train a joint representation. And also rare species are the one that benefits the most from the shared model. The gains are not huge. I mean, this is still a problem where deep learning and AI in general is not, I mean, it starts to be shown that it's better than classical machine learning, but still we are very close, right? Yeah, but still we did show that, yeah, with deep learning backbones, you can encode the spatial structure that we could not do with another model, and so you can improve performance of that. You can also benefit from the temporal structure, so like using a time series of satellite imagery. This gives you information about the phenology of the habitat, and so then your prediction of what species is there is more accurate than just using a single image. And yeah, generally, yeah, those deep learning models are very good to extract some relevant ecological information from any raw data. So this is an example where you train only a model to predict species based on satellite images, and so you can like create this t-SNE visualization with the images, and then on the bottom left you can color each dot by the average temperature of the location where the satellite image was taken. Okay, and what you see is that there is a clear gradient. You see that the principal component of this space is the temperature, actually. So that means that the model is able to learn the temperature just using the satellite imagery. So of course, ecologists will say, okay, so just please train a model on the temperature. All right. But yeah, still it shows the powerness of the thing. And yeah, and so this choice of modality is then crucial when you train a species distribution model. So what to use as inputs. So this is an example where you start with a model only with Sentinel-2, and then it's an ablation study. You add a new variable and another one and another one. So yeah, you increase your performance, you are happy. But then you remove the Sentinel, which you expected to be the best one because it's satellite image, you're happy. And then, oh, surprise, okay, it's almost the same performance without the Sentinel, right? So yeah, in terms of information theory, it's because, yeah, what you expect is that each modality brings a new piece of information about the species. So the yellow region is the information of the species, and so you expect each modality to bring some new information about that. Okay, so this is the information contained by X1. This is the information contained only by X1, okay, which is different. This is the information that is common to the three modalities. So if you have either one of the three, it's sufficient, okay? And so in the ideal case, each modality has some specific information that it only encodes, so you need the three modalities to get the final result. But sometimes it's more like that. And so what you see here is that the pink modality is very informative. It's the one that captures the most, but it has no specific information. So all the information it captures can be captured by another simpler variable, okay? And so this is some more what happens to the satellite stuff. Yeah, so to work on all those topics, we're organizing a challenge which is called GeoLifeClef, where we try to build models based on those different modalities. So in this context, we are sharing a data set and some baseline models so that have been published in an ERIPS paper. So you see on the right a model where still the three modalities are complementary, but the satellite one is the one that brings the less information, and the two other one are climatic cubes. So it's just a temporal series of the temperature and precipitation that we put in the shape of a tensor to do some features engineering to help the model train on that. But the most informative modality in this case is the Landsat cubes, and the Landsat cubes, it's also a time series of 6 bands of spectral values, And so this really gives an information about the history of the habitats at a given location. And this history, of course, matters a lot of what you observed in the end, because like a forest takes time to grow. And so if you have the history of all what happened during the last 20 years, this gives you a lot of information. Okay. So this is about the X, but here I'm going to talk a little bit about the Y, so the output, because there are different tasks you can solve. The ideal one is abundance data. So you have your training set is abundance data. So you know for each species how many individuals you have. And so that's the richest information, but it's very, very costly to produce. Then you have presence absence data, which is the exhaustive list of species present. So it's also hard to produce. And then you have presence only data, which is typically what we collect in ntNet, which is, poor data. I mean, it's only one label and it's biased because it's not just a uniform sampling of the presence absence. People decide to look at the species more than another one. And so you have a lot of data like that, but they are highly biased. And but you have to deal with it. And one thing you can do, which is quite straightforward, is like you pre-train a model on those PO data because it will capture the relevant variables and things like that. And then you will fine-tune on your small data sets where you have richer information. We did that for fishes, so in a paper in Ecology Letters. Another things you can try, and that was doing by ecologists, is to try to capture a relative probability of the species across the different sites. And we extended this model that is very popular in ecology for deep learning. And so it appeared that the function they are using, we see on the right, which is a normalized intensity of the species by a partition function. So if you extend that to deep learning, it's just a normalization over the batch. But it's a column normalization. So meaning that contrary to usual, like a cross entropy where you do a softmax on row, here you do a softmax on the column, okay? So for each species, you do a softmax across the sites that are in the batch. So very simple, but this allows to improve, like the distribution of each species individually. It does not improve the assemblage if you look at like macro average metrics, or it's the opposite. It does not improve sample average metrics, but it improves macro average metrics. So still in those models, you always have the hypothesis that the species are independent, okay? And so another key thing, I think, for the future is to try to capture the interdependencies between the species in an assemblage. And because, the Y is also a complex signal. So if I go back to my information theory interpretation, each species share information with the other. And so some are predictive of the other. And so you would like to capture all those shared information. And the first thing we are currently looking at is just an encoder of such signal. So how to encode that. So since it's tabular data, the first things we were trying is like normal machine learning models, or models like tabNet. I don't know if you know, there are some transformers for tabular data, but since there is a lot, I mean, this vector is huge, it's 10,000 of species, and so that was not working node. And finally, the idea we had is to only encode the present species, so only take the 1 and not the 0, okay? And and tokenize that. And so if you think at it, it's like a sentence of species. So you take the sentence of species on the location. So they are sorted by decreasing order of dominance. And so you have a sentence, and we are training some masking language model. So that's why we called it ntBERT. And so the idea is that you predict a species that has been masked in those input sentences. And so this really allow to learn the syntax of those species assemblages. So how are species dependent between each other? So it has been trained on a big data set of 1 million plant species assemblage, so presence absence data. Okay. So what we can see, so here it's the performance on the task of predicting the missing species. So you can see in blue that it's, oops, sorry. It's like 17% accuracy to predict the missing species. It's much, much, much better than just using the co-occurrence information. So if you just use, I mean, yeah, that's what ecologists do. Usually they take co-occurrence matrix, and it doesn't work to predict the missing species. It's patterns that govern really the fact. It's not only co-occurrent. And if you compare to like a normal neural network with binary cross entropy or things like that, it's also much better. And interestingly, if you look at the red curve, it's better on frequent species. So the most dominant species are easy, but it's just failing to predict the others. But what you see with the plant BERT model is that once you have captured the three first species, sorry, you have sufficient information about the habitats to start predicting better the remaining species. So that means that it detects this pattern of this combination of species, and then it improves the rest. Yeah, and then what we did is to fine-tune this model to predict habitat labels. So habitats are also an important information that ecologists want to monitor in addition to species. And so, yeah, what we did was to predict the habitat from this species list. So the plant BERT model is first pre-trained on the masking task, and then it's fine-tuned to predict the habitat. And it performs very well on this task. So it has better prediction performance than other models that are using not only the species, but also information about the environment. So here we did better only with the species information. Yeah, and if we put all those models in one tool, which is called GeoPl. So this is a mapping tool we are developing in PlantNet. So it's like the new software of the PlantNet family. Yeah, so we created maps, high-resolution maps of species and habitats at the European level, so at 50.50 meter resolution over the whole Europe. So it's 4 billion cells to be inferred. So for each cell, we will extract the cubes I was talking about, do an inference. This predicts the set of species, and from the set of species, we can compute indicators about like number of protected species, number of local species, number of invasive species, taxonomic diversity. And then we also apply the plant BERT model to predict the habitat, so for each cell. Yeah, so you can try the tool. It's on geo.plantnet.org, and you can explore all the species maps that are in Europe. And yeah, you can also do some polygon queries to get some reports about a particular area, which would give you how many protected species and things like that. So it's kind of digital twin of the plant biodiversity. So of course the models still make errors, and so it's like a first version. So in this first version, there is only, unfortunately, the satellite branch, so the one that was working the less in the three. But we are currently producing the new maps with the 3 branches, so they should be integrated in January or February, I think. So the model is already there, and it's already running. Yeah, so that's it. I will present some other stuff on October 23 at the IRBV lab, so maybe more on the applied side and about plant biodiversity monitoring. And yes, thank you very much for your attention.
For this great talk. I'm sure there's lots of questions, and there will be some questions, I'm sure, online as well. So you can. So, any questions for Alexis from the audience? Guillaume?
is your application supporting somehow the growth of the users? Like, I'm not using ntNet, but I'm a user of iNaturalist, and let's say I snap a picture of a swallow, and I say it's a swallow, but I don't know if it's a European swallow or an African swallow. So I just say it's a swallow. Yeah. You know, the key is a component of that. And a few hours later, 3 people tell me it's a European swallow, but I'm just there with my picture, and some experts telling me it's a European swallow, but I don't know why. Next time I snap the same picture, I haven't evolved as a biologist or as an amateur iNaturalist to say, okay, now I'm sure this is a European swallow. So is there anything in that that will help my growth as a naturalist?
Sorry? Yeah, so yeah, I mean, the question is whether we are doing something to allow people know more about the species and progress on this knowledge. And so we have another software that I didn't talk about, which is called The Plant Game, and it's really an application that is dedicated to train people and to progress on that. There are connections with PlantNet because in this tool we are really learning the confusion matrix of the user, so the entire confusion matrix, so to know where they make errors, where we should make them progress, that's we will probably try to integrate back that in PlantNet at some point, but so far it was a separated tool and only working on French flora. But this is clearly topics we are thinking about, how to make people progressing. So this is the first part. The other part is more, the next step will be multimodal models, I think. So we really want, I mean, what we would like is a system saying, okay, now you have 2 possible species, please take a picture of that, and you will disambiguate them. I mean, that's really where we want to go in the future. So like, maybe chatting with the AI, and he would say, okay, you should look at that and that, but this requires, of course, learning models on all the knowledge about one species and things like that. Yeah.
Yuri and then Laura. Just a question regarding the distribution models that you've played with now. You showed that after removing Sentinel wasn't really that impactful, but how do you prevent the model from accidentally learning incorrect information from the features that you provided? Because sometimes if you train a model to do X, it kind of learns that the shadows might be important, then it makes false predictions, something like that. How do you kind of force the model to really extract the meaningful features from the data set that you provide?
Yeah, I mean, the keyword is diversity in your data. So I mean, you need huge amount of data, but the most important thing is to ensure you have diversity in your data to avoid overfitting and such things. So we don't do yet something special on that, but we are always careful about potential bias you can have in the data, and when you evaluate, always have splits that you are sure so like special block holdout things, or be sure that your split between training and validation is something that has a sense.
Laura, your question?
Yeah, thanks for the talk. Really. I have a question about kind of going back to user experience, and we built tons and tons of SEMs, and you give it to an expert, or an expert can look on the platform. And I would say almost as a rule, the model that predicts the best according to our evaluation metrics is never the one the expert likes the most. And particularly as we get more of these like machine learning models and deep learning models, and you do all this fancy stuff like borrowing strength, like the more you go down that road, it just seems like they just don't like it. But have you like had any like of the app that you have? Do you go back and forth with the user a little bit in that way, and do you have a solution?
I mean, we are. We are interacting a lot with experts in conservation. So it's developed in the context of those 2 European projects, and so one of them is more on the research, and the second one is really on decision making and things like that. So So yeah, I mean, I will not say they prefer. In our case, usually they prefer the best performing model in what we evaluated, but yeah, I mean, it depends, and we have to continue working with them. But yeah, of course, they see the errors, and I mean, they help us to better understand why is there errors at this place or not. But yeah, if I go back to PlantNet, I mean, for the species identification 10 years ago, they were the same problem, and because the model was bad. I mean, and then now it's good and they are all happy. So I would say we still need to do some research to make the model better. And then I'm pretty sure if it's really better, because here it's better, but at one point, I mean, we need some big gap somewhere. And so we are trying to understand what is missing. So is it only higher resolution and to really see the species, or is it, yeah, but I mean, we are happy to see that this temporal dimension was really bringing something. So probably there are other modalities that could really help and really work. And also the models themselves could be better. So we will see with the V2 version, so the 3 branch, we will probably have a new answer. I mean, if they prefer the V1, I will be, I'm sure they will prefer the V2, because yeah, the performance is much better. And we know the V1, for instance, it's like confusing beaches with snows, or yes, and just its image, you know. So with the new one, it should really remove such confusion. So I really hope they will like it. We'll see.
Thank you so much. So it's already 11:30. However, there is lunch. I don't think it's for everybody. It's late, but yes, there's enough for everyone. Okay, right. I didn't want to make sure that the speakers are starving or something. So there is some time left if you want to stick around, have some food, talk to Alexi, other people. But again, thanks so much, Alexi, and yeah, it was great.
have no question, but you have people online who are saying nice presentation.
Thank you.